I've spent so much time watching theoretical science like physics or math just shift on a dime. A new concept comes out and everything changes.
Right, a single paper can rewrite the field.
Exactly. But biology, that's always felt different. That's the wet lab. Just
Right.
a grind.
Oh, it's a total grind. All that pipetting, waiting for things to grow. The iteration cycle can take weeks, even months.
And that's the bottleneck, right? That slowness.
It absolutely has been. But that is precisely the bottleneck that, well, it looks like GPT-5 just shattered.
Okay, so let's get into this. We're doing a deep dive on an evaluation from OpenAI and Red Queen Bio. Their mission was pretty foundational, wasn't it? They wanted to optimize a key tool.
A really key tool, the HiFi molecular cloning protocol. Basically, think of it as the best way we have to cut and paste custom DNA sequences together. They wanted to make it way more efficient.
And the headline result is it's kind of unbelievable. The AI proposed changes, totally on its own, that led to a 79 times improvement in cloning efficiency.
79. It's not a small jump. It's a different league. That's what's so incredible here.
So where did that huge gain come from? Let's break it down.
Well, the first big piece, the primary enzymatic leap, was something brand new. The AI basically invented a protocol it called RECA-assisted pair and finish HiFi assembly. Let's just call it RAPF HiFi.
And that new process alone was a big deal.
It was. That part by itself gave them a 2.6-fold gain.
So what was the mechanism? What did the AI figure out that was so novel?
This is where the reasoning gets really complex and, frankly, impressive. It wasn't just adding one thing. It added two new proteins.
Okay.
First, a recombinase from E. coli, kind of re-EA. You can think of it like a guide. It searches for the matching DNA pieces and sort of pulls them together.
A biological GPS.
Exactly. But here's the problem. Re-A is notoriously fussy. It gets inhibited by tangles in the DNA, what we call secondary structures.
So the AI anticipated that problem and solved it at the same time.
Precisely. It paired RECA with another protein, GP32, and this protein acts like a tiny biological comb. It just smooths out and detangles the DNA, clearing the path so RECA can do its job.
Wow. So it's not just adding ingredients, it's understanding the chemistry, the friction between them.
And the timing. It also built in this complex multi-step temperature cycle. It goes to 50 degrees, then drops to 37 for these new proteins to work, and then back up to 50 to finish. That specific instruction, that was completely new.
That's amazing, but that's just a 2.6x gain. The rest of that huge 79x boost that came from somewhere else.
It did. It came from optimizing the downstream process, specifically the transformation step. This part alone yielded a massive 36-fold improvement.
And what was the trick there?
This is where the AI really challenged, decades of human lab wisdom. The change was simple, but so counterintuitive. It involved pelleting the cells, which are super fragile, and concentrating them at a cold 4 degrees Celsius.
Which is something you're basically taught never to do.
You're taught to handle them like they're made of glass. But the AI figured out they could tolerate it, and that higher density just massively boosted the success rate.
It's incredible how an AI can find these solutions we've overlooked because of our own assumptions.
Yeah.
But to actually run these kinds of tests, to iterate this quickly, you need some serious automation and infrastructure.
Oh, absolutely.
And on that note, if you or your business needs help training AI models or implementing automation in custom software, or even just figuring out where intelligent agents could make the biggest impact, you should check out Embersilk at embersilk.com for all your AI needs.
And speaking of automation, that's exactly how they proved this out. They used a robotic system, robot on rails, to execute these really complex protocols. The robot did all the work.
And it worked. The robot got the same result.
It got comparable results, yeah. It achieved about 89% of the improvement that the human researchers did, which is fantastic. It absolutely proved that the AI's instructions could be validated and run autonomously.
So what does a 79x acceleration like this really mean for science, for the world?
I mean, it means the whole research loop just got compressed from months into days. These new mechanisms, like RAPF HiFi, they're likely modular. You can probably repurpose them for other things.
So it's not just one trick for one protocol.
Not at all. This gives us a really hopeful vision for the future, A future where biological discovery moves at the speed of computation. It could dramatically lower costs and, more importantly, accelerate how quickly we can turn these discoveries into real-world applications that help people.
A fantastic and really optimistic look at the future of research. If you enjoyed this deep dive, please subscribe to the show. Hey, leave us a 5-star review if you can. It really does help get the word out. Thanks for tuning in.